Every year, 500,000 young Canadians, as many as play hockey, participate in school science projects. Last May, the largest Canada-wide science fair ever held was hosted by the University of British Columbia in Vancouver. With finalists from all 10 provinces and 2 territories, a total of 382 projects were displayed by nearly 500 students. Of the 91 projects in the senior division, 4 were selected to receive the coveted Manning Young Canadian Innovation Award.
I think innovation is something that we can all do, and it just takes one spark, one idea, and one passion. And it leads you to these amazing things as long as you're able to communicate with others and bring the ideas of others together to create great strategies to help mankind.
It's a pretty good feeling being an innovator right now. I actually really enjoy helping people.
I think that's basically what it's about, is being able to look at things in a different way or frame a problem in different ways so that your solution turns out to be something unexpected or something that's not too common.
You need to continue evolving the technology and make it better and better and better as you go. And that way you can solve all the problems that keep coming up.
We need science in our world to survive. We take it so much for granted nowadays because our whole world is based on science. But we all need science. We could do nothing we do today without science of some kind.
Hi, my name is Jennifer Wong, and my project is Rooting for a Cure.
A casual observation led to a startling discovery for Jennifer Wong of Saskatoon. Jennifer noticed a relationship between the wild sarsaparilla that grows in northern Saskatchewan and ginseng. Her curiosity led to a full-scale study to see if the local plant would also yield medicinal herbal qualities. Jennifer's surprising discovery: a new sarsaparilla extract that kills cancer cells while leaving healthy cells untouched.
I actually went up to Laurent myself and collected these plants and then divided them into 4 parts, so the leaves, the stems, the roots, and the berries. And then from each of these 4 plant parts, I extracted 6 extracts depending on chemical solubility. And then so I had a total of 24 extracts to test against cancer cells. And then from that I took the best chemical properties from this plant and tested them against normal cells to see whether it'd be able to differentiate between healthy cells and cancerous cells. The results were that not only did the mortality rate of cancer cells, it was almost 100% consistently, but when I tested it against normal cells, it required a much, much higher concentration to actually kill off any normal cells. Not only will it be able to target cancer cells and kill them off in a patient, but it'll be able to maintain the normal cells and make sure that people's immune systems aren't too crushed. I worked on this project for about 8 months continuously, so it was a very long-term project that I had to dedicate myself to. But then to get an award like this, it's so big and really, really exciting.
My name is Chet Gervais, and my project is the Matrix Probe Evolution.
Chet Gervais of Amherstburg, Ontario, worked part-time for over 2 years in a breast cancer screening clinic. Chet grew dismayed by the potential for error with standard testing methods. If only a more accurate breast cancer detection test could be added, lives could be saved. Chet conceived a unique multimodality fusion breast imaging system, combining digital mammography and 3D breast ultrasound that promises improved diagnostic accuracy.
I'm basically out to improve the misdiagnosis of breast cancer because right now, having the range of error over 25%, 1 in 3 women are being diagnosed with breast cancer, so there's quite a lot of improvement to be done. My future application is actually a matrix, hence the title, or a series of piezoelectric crystals, or just crystals which produce ultrasound. Now they're in sequence, and due to electrical stimuli or electrical impulse being sent into them, they basically fire off in sequence, giving you 100% view of the breast tissue. And that's compared to the 1 to 2% of the breast tissue actually seen today. The experience of being an innovator is actually a very good feeling. In this case, the help of diagnosing breast cancer in female patients. I'm very excited about this because this could actually go very far.
Hi, I'm Patrick Danielson.
And I'm Robin Miro.
And this is our project, Macroinvertebrate Sampling Technology, aka MIST.
Growing up in North Bay, Ontario, Patrick Danielson and Robin Mirren naturally love to fish. But the fishing hasn't been that good lately. Microinvertebrates from Europe have stormed our lakes, upset the lower food chain, and denied the fish population their natural diet. While the boys had no way to curb the effects of invading organisms, they did have a way to help the researchers who could. Their invention? A boat-drawn device to detect the invading microinvertebrates and precisely map their location on 3D charts.
Basically what we did was create a detector that in real time on a 3D plot of a lake shows precisely where macroinvertebrates are located.
What we do is we drag it underwater behind a boat, and what happens is the species will flow through it, and our detector is actually hooked up right here.
They're going to flow through one of these holes, and basically what happens is we have a sensor in here. Whenever something breaks the beam in there, it analyzes how long it is and how transparent it is, how much the light goes through it. So from there, you can tell what kind of species is flowing through here, and then it cross-checks with a global positioning system, a GPS, to show you exactly where in the world that particular species was. So pretty much the little macroinvertebrates, they can run, but they can't hide. We can always find them with this detector. So it builds. You have the healthy aquatic ecosystem, and if your lake's healthy, then you have healthier fishing. If your fishing's a lot healthier, then there's more tourism, and we can actually prosper in Northern Ontario.
Hi, my name is Carrie Williams, and this is my project, 2378 tetrachlorodibenzylpdioxin, targeting toxins.
Growing up on a ranch near Merritt, BC, Carrie Williams experienced firsthand a dioxin scare. Toxins were entering residents' bodies by way of the feed given to their livestock. Carrie started reading up on dioxins, often called the new asbestos, for the time-delayed effects they can cause. In conclusion to her literature review, Carrie presented her findings to help tame the effects of dioxins at the Canada-wide Science Fair.
These nasty chemicals get inside our immune system, inside the fatty tissues within our bodies, and cause things like birth defects, cancer, and premature cell death, and that's definitely something we need to address. So what I'm trying to do is, when they are in our bodies, is actually prevent the toxicity, which we know is occurring, from happening, and also limiting them from initially getting into our bodies. Well, basically what I've done is I've done a literature review in which I've pulled sources on how dioxins, specifically the 2378 tetrachlorodibenzylpdioxin, interacts with receptors as members of the tumor necrosis factor family of receptors. I also looked at how these two receptors, the CD95 receptor and the aromatic hydrocarbon receptor, interact with dioxin-like compounds. By doing a statistical analysis, I was able to relate those compounds to the dioxin and therefore develop the pathways through which the dioxin causes toxicity within the human body, and also develop solutions by analyzing those pathways. I am 17 years old.